The nervous system module of the pure MedSim simulator. No other simulation system can claim anything of this kind. The nervous system module may function as a separate subsystem. At the same time, it performs regulatory actions when the system is working as a whole. The nervous system mode is accessible when the brain as the subject of studying is selected in the main menu. Having initiated this mode, a user will observe windows with a concept map of the central nervous system, with main parts thereof highlighted in blue, and the map of the central nervous system's peripheral part, where blue color shows names and numbers of spinal roots. The principle of operation of this mode implies left-click selection of a part to be studied, with further clicking through particular sensory and motor areas, Brodmann areas, correlated to diverse cortical functions. Let us see how this mode works with several left cerebral hemispheres areas taken as example. First, we need to left-click on a corresponding term highlighted in blue on the CNS map. A window with two images of the left cerebral hemisphere will appear. The upper image is a dorsal view of the hemisphere. The lower one shows a cross-sectional view. Digits stand for various sensory and motor areas. When a mouse pointer gets within an area's borders, such an area will be highlighted in green. A left click on the area highlighted confirms selection. As an illustration, let us place the pointer on the motor area exerting control over mimetic muscles. There will appear a map of neurons associated with this area. Please note that an image of any separate neuron found in such maps of neurons corresponds in fact to neuronal pools exerting control over muscles or receiving sensor signals from receptors. Neurons from this area manage contraction of mimetic muscles. A left click on the image of any neuron shown on the map will result in contraction of a corresponding muscle. Let us see what happens if we left-click the leftmost neuron. The neuron selected will be highlighted in red, and the diagram above images of neurons will show action by potentials generated. An orange line on the CNS map will trace the path through corresponding areas. A red line on the CNS's peripheral part map highlights the path of axons leading to corresponding peripheral nerves. A scheme of mimetic muscles innervations will appear with a red line tracing the tract's peripheral nerve. There will appear the simulator's head, demonstrating corresponding contractions by means of mimic changes of the particular part of the face with the acting muscle highlighted in red. Let us see how it all works once again. Let us take another example of managing motor activity, say in the lower body parts. First, we select the left hemisphere on the concept map of the central nervous system. And then we choose in the hemisphere scheme the area exerting control over motor activity in the lower body parts. There will appear a corresponding map of neurons. A left click on the lowest neuron will highlight it in red, and the diagram above images of neurons will show action by potentials generated. The screen simulator's leg will move accordingly. Another example deals with a sensory area functionally related to pain sensitivity. First, we select the left hemisphere on the concept map of the central nervous system. And then we choose in the hemisphere scheme the pain sensitivity sensory area. There will appear a corresponding map of neurons. Let us see what happens if we left-click any neuron. The neurons selected will be highlighted in red, and the diagram above images of neurons will show action by potentials generated. A corresponding path will be shown on the screen simulator, and the zone innervated by the neurons selected will be highlighted in green. After the facial skin pain sensitivity area has been selected, there will appear a window showing a neuronal pool managing the corresponding innervation zones. A left-click excitation of any neuron, which will be highlighted in red, produces an image of a particular nerve highlighted in lilac, and a respective innervation zone highlighted in green. Let's do it again. After the gustatory cortex has been selected, there will appear a window showing a neuronal pool managing the corresponding innervation zones. A left-click excitation of any neuron, which be highlighted in red, produces an image of a particular nerve highlighted in lilac. A respective innervation zone on the tongue will be highlighted in green with indication of the corresponding type of taste. Another unit of the nervous system mode deals with eye movement and image control. To see how it works, first we select the left hemisphere on the concept map of the central nervous system. Then we choose in the hemisphere scheme the part of the primary motor area associated with eyes movement. There will appear a corresponding map of neurons. Let us left-click the leftmost neuron, for example. The neuron selected will be highlighted in red, and the diagram above the images of neurons will show action by potential generated. The screen simulator's eyes will move accordingly. Now, keeping the window with the map of neurons open, we will select the visual cortex in the left hemisphere scheme. There will appear a window showing the image formed when the simulator's eyes are looking straight forwardly. If we now repeat above actions with neurons to make the eyes move, we will see the images thus obtained shift correspondingly. Let us see how the vegetative nervous system works with the respiratory center, taking as an example. The respiratory center is the center in the medulla and pons that determines the signals to be sent to the respiratory muscles, thus regulating the rate and depth of breathing. When the medulla is selected from the menu, there appears the image of related clickable areas highlighted in green. The respiratory center 1 controls the rate of breathing. The respiratory center 2 controls the depth of breathing. Inspiratory and expiratory centers provides synchronous automatic generation of electric potentials driving the diaphragm. Once any area is selected, there will appear the respiratory center window. The rate of breathing can be changed by left-clicking on one of 3 neuronal pools on the top right corner of the window. A normal rate value has been set by default. When the respiratory system mode has been selected from the main menu and, for instance, the lumen of bronchi is reduced, G carotis will react to changes in CO2 and pH levels, which will lead to automatic alterations in the rate and depth of breathing. By the same procedure, in all scenarios resulting in changes in CO2 and pH levels, the respiratory center will react automatically. Let us see how agnostic areas and areas related to praxis, body schema area, writing area, Broca's Wernicke's area, work with walking, taking as an example. This can be done as follows. First, we select the left hemisphere on the concept map of the central nervous system. And then we choose in the hemisphere scheme the area associated with body schema. There will appear a sub-window of the neuronal pool with neurons memorizing and triggering sequences of muscle contractions resulting in various movements. The neurons are colored according to the strength of related muscles contraction. Let us go through the steps needed to get the left leg move. Now we will have to get the other leg move. Same actions as above, save for selection of the other hemisphere, are required. The next step is to select the left hemisphere on the concept map of the central nervous system, and then choose in the hemisphere scheme the area associated with praxis. The neurons will be excited in the sequence set before, and the simulator will begin to imitate walking, moving the legs alternately. A window with a scaled-down simulator image will appear beside, with arrows indicating leg movement directions. Closing the window with a scaled-down image stops leg movements. To study principles of vision, first we will have to set the image formed when the simulator's eyes are looking straight forwardly as described before. The next step is to select the eyes mode on the concept map of the central nervous system. That will invoke the visual system window. Visual fields are located at the top of the window. Each segment of a field corresponds to a neuron rendering image to a particular part of the brain. The red line outlines the field of view. Left-clicking on segments within the zone outlined, selected segments will be highlighted in green. We'll mark them as those with optical defects, faulty paths traced by colored lines for corresponding eyes through retina and nerve tracts. The visual defects will as well be revealed on the image a user has set. After the stereognosis area has been selected, there will appear a window showing neuronal pools highlighted in various colors. Each color corresponds to a particular muscular effort in the process of writing. If we select the writing area now, there will appear a window with a corresponding neuronal pool. A left-click excitation of the pool will produce a window showing a word written in accordance with the color pattern of the neuronal pools. In the same window, a user can draw any image. The color pattern of neurons will change accordingly. Now, excitation of the neuronal pool will command the simulator to draw that new image again. When the area associated with speech production is selected, and the corresponding neuronal pool triggered, the simulator will utter a phrase set by default. Pure MedSim Life Simulator greets you. The Vasomotor Center provides vascular tonus and heart rate managing. It is initiated automatically when the system as a whole starts working. The center's functioning can be studied separately when the medulla is selected on the concept map of the central nervous system. The corresponding window features areas of respiratory and vasomotor centers. The Vasomotor Center area is located under the images of inspiratory and expiratory centers. Left-click selection of this area will invoke the Vasomotor Center window. The left neuronal pool exerts control over vascular tonus, and the right one manages heart rate. Control is exerted automatically when corresponding scenarios have been selected, bleeding for instance, or when other interactive actions in the simulator modes, for example hemodynamics, are taking place. This module will be further developed to assist scenarios related to CNS pathologies, strokes, brain tumors, etc. The corresponding areas will be damaged, which will be reflected in symptoms revealed both by the screen monitor signals and by the virtual patient verbally.